The goal today is to explain this new deep learning phenomena, the core philosophies and building blocks, what some of the results that we've gotten out of my lab, and kind of the outlook of where this is going in the field, and especially for this community, what it means for biophysics. All right, so let me start with classical protein structure prediction and design. That's the same as the title up there. You know what? We can do a little better on this camera here. I like to use my hands, and I know the board has a little bit of a delay. So let me know if I need to clarify anything, and I can also take questions anytime. All right, so in classical protein structure and prediction and design, again, we're taking this sequence, this floppy piece of spaghetti, and we're looking for the fold, assuming it's a well-folded protein. That's a three-dimensional structure that dominates the partition function that has the lowest free energy that would be observed most of the time in nature. And we think about this, that the sequence determines the structure. And then, of course, the structure determines the function. So if we have the structure, that's good, because then we can say something about what it does. Let me go this way. That way, there we go. When we do this classically, there's two core challenges. The first is the search challenge, in that we have to search all of the conformations. And the second is the scoring challenge, or the energy challenge, that we have to score each of the conformations that we generate to figure out which has the minimum energy. Of course, this whole thing we could run in reverse also for the design part of our field, where you have some function and you think about what structure of a molecule would perform that function, be it catalysis or binding or blocking or something like that. And then the structure, you would say what sequence is going to fold into that structure if you're doing design. If you're doing the design, then we're going to search not the conformation, but we would search the sequences that would fold into a particular structure. So this is a hard problem, as all of the first-year biophysics students know, because of Levinthal's paradox. Levinthal, who said each amino acid could be alpha helix, beta sheet, or something else. So that's 3 possibilities for each amino acid. If you have 100 amino acids, that would be 3 to the 100th power, which gets really big. That's too many conformations to search. And then the scoring, this rests on Anfinson's dogma. Again, the first-year biophysicists will remember, Anfinson found that if you unfold a protein by heating it up and then cool it off and refold it, it's active again, at least for biophysical proteins that behave that way. So that tells you that you don't need a ribosome or a particular folding pathway. Apologies to Stephen Fried, who I know is working on finding out when that's not exactly true, but in a general, biophysical protein will fold back and have its function. So really, if you knew a thermodynamic energy function, you searched lots of configurations and you calculated the energy of each, you just pick the lowest energy conformation, and that's what you should see most of the time in solution. Again, assuming it's one of these proteins which has a single state that dominates all others, that dominates that energy landscape. So in this scheme, and we've been pursuing this in my lab and all of the Rosetta labs and lots of protein folding labs and protein design labs. In this scheme, we start with postulates. Is that coming out okay? And theory, like what are the intermolecular forces for those energies, and what different geometries are possible. And we use those to then make predictions of structures, energies, maybe we get implications from that. So we predict some kind of structure from that, from these first principles of, here's the van der Waals force, the hydrogen bonding energy, and here are some structures that we zoom through knowing them from proteins. If I back up a minute, these postulates of van der Waals energy and hydrogen bonds and solvation forces, those originally come from data and experiments where people look at what's the vapor pressure of hexane. And so what are those van der Waals constants? And you get those data and then you build these theories and then you make predictions from that. So it's kind of a bottom up and it has a structure to it physically. All right, so the deep learning era is a little different. This is a machine learning or deep learning paradigm. So the deep learning grew out of the machine learning community. The deep learning is a bigger, deeper model, and I'll show that in a minute. But the idea here is you just get the machine to learn and you start with data. So you simply start with inputs and outputs. And then in between, you stick a model. And you want your inputs go through some model to give you the outputs. And then we're going to train this model by going back and forth, taking the outputs and going backwards and seeing what inputs and going back and forth until we get this model to connect the two. In some ways, that's like what we did here in the classical, because we did experiments and came up with van der Waals constants and charges, and then we tried it and we looked to see, does it predict structures, and we found it didn't, and then we went back to more data and back to more testing. So we did this, but it was a very manual process over many years and trying to refine the rules of physics, like the 612 potential for van der Waals approximation, or Coulomb's law with screen charges for electrostatics. In this case, we just put the inputs and outputs. We use a lot of data, and then we let the computer go back and forth and train this giant model. So the example here, the input, of course, would be the sequence of a protein, and an example here is the output could be the structure. So we'd map the sequence to the structure, and the model is essentially a giant curve fit, if you will, between these data. Y equals f of x, where f is some giant function. X is, I'm going to underline it to indicate a vector, or in this case, it could be a general tensor, a lot of data, like many, many x, y, z coordinates for the structure, or for x, the input, many, many amino acids. In the machine learning lingo, the inputs are called features, and the outputs are called labels. So you start with a set of features and you predict what label would come out of it. Here's a picture. Should we label that picture a cat or a dog? That's an example of putting a label on it. A lot of the work in my lab over the last 2 years has just been translating the jargon from the computer scientists into what we're doing, and my students have helped a ton with that. All right, what else? This is structured with a layer of neurons like your brain, and the idea is our brain is a great pattern matching machine. We see patterns, we match them, we try to make patterns and give it structure, and that's what we're going to try and do here, is what is the structure of the data that's going to map sequence and structure. Okay, so the next big question, once you have this, is in our problem, what do we do for the inputs and outputs? In the machine learning community, what this is called is the representation of the data. So somehow we have to encode.
Jeff, I think you're outside the border.
Okay. Oh, I so am. Yes, thank you. Okay, time to erase and go back. They built these giant boards. Actually, some rooms have these tiny boards, and then you have to erase every 2 minutes. Oh, and the eraser's not working so good.
The eraser's coming up okay for the screen, at least.
Okay, thank you. All right, so the first thing when you say I want to do deep learning with with proteins is you have to say, what are my labels going to be? What are my features going to be, right? So the sequence is a string of letters, but a string of letters is not, I mean, you can digitize that, but there's a simpler way to do it. So what we're going to do with that is what's called one hot encoding, where you are going to take a series of bits. For an alanine, you're going to have 20 bits. For an alanine, you'll put a 1 in the first slot and then 20 zeros. And for a cysteine, you'll put a 0 in the first slot and a 1 in the second slot, etc. So we would basically have a binary code, 1 or 0 in each of 20 slots to indicate each amino acid. So that's called one hot encoding. For the structure, the thing we'd usually look at is X, Y, Z coordinates. You can do that. The challenge there is that X, Y coordinates are not unique. For one thing, you could rotate the protein around different ways or just translate it. You have the same fold. So what you want is a 3D invariant representation. There are some various ways of doing that. My favorite, or the simplest and intuitive to understand, is instead of having absolute X, Y, Z coordinates, you'd use distances between points within the structure. So we do this with a pairwise distance matrix. All right, have residue 1 to n and residue 1 to n. There's a diagonal there. And at each point, this would be a big matrix, and at each point you have a number between the two C beta atoms. Seems to be what works well. So for example, if we had an alpha helix, you have all those I to I plus 4 contacts, and you'd see a set of numbers be large there along the diagonal where I to I plus 4 distances are short. If you had a strand, you'd have close contacts close in sequence, and then they'd get progressively far away, and you'd see a band that looks like this. This matrix is symmetric, so I'm going to draw it that way too. And so you can get these pictures of the distances between all the points along the protein. If these things connect later, and maybe you have a long-range contact here between the N and the C terminus, that would show up way off on the diagonal. So those are harder contacts to find. They're not local pieces, but they would show up in this image. All right, so these are the most common representations. There's several others. For features, sometimes people will take, say, the hydrophobicity of a residue, read that off a scale, or the size of a particular residue, or the charge, or even a predicted secondary structure. So you could give those as numbers and feed those as inputs, because those are easy things to calculate. For outputs, another interesting output is a shape descriptor, or a surface descriptor. And I won't talk about this today, but there's some really great models where it looks at a surface and an interface and describes what's on that interface. Any questions about the representations? All right, I want to dig into the math next, because
Stephen had a question.
I had a quick question. So the sequence would be a 20 by n matrix, right?
Exactly. A sequence would be, yeah, would be yes, 20 by n, 20 bits and n residues long. So it's actually a matrix of numbers that we'd be feeding in. All right. So this is a chalk talk. We're supposed to do some basics, and I'm an engineer, so we're going to dig into the math a little bit so you see what goes on in the inside here. Okay. So one way to think of this is you're going to put these inputs, and the model has to sort out the inputs between the different kinds of proteins, the different structures. So it has to learn from them and categorize it. So you could think about it, if this is protein fold space, which I made as a box, which is ridiculous because it's a huge space, so it's not really finite, but you know, you might have some alpha helices in this space or some helical bundles. So each point here might be a different alpha helical protein. And then there's other protein folds that might look like beta strands or IgG folds. And so you'd have a bunch of beta points over here. I don't know if those are showing up. And then you might have some mixed alpha beta in another region. So one thing you can think about this deep learning when it's trying to learn what does a protein look like, it needs to look at a lot of data and a lot of examples, and then it needs to separate them and learn the differences. So one simple thing it might learn is it might learn the boundaries in protein fold space between the alpha folds and the beta folds and the mixed folds, right? So you want to separate these things. So all right, we had that paradigm and I had y equals f of x. I want to construct what that function f is. If I want to separate different things, one way to do that would be with a function that looked like that, that goes from 0 to 1. This would be a sigmoid function. And the sigmoid function sigma of x equals 1 over 1 plus e to the minus x. So what's nice about that, let's see, the midpoint here is at 0, and it goes from 0 to 1. So if my output is going to be, is it an alpha helix or a beta strand, this function could do it. I could have some inputs, and then I could say either it's, my inputs are here, it's alpha, and if my inputs are there, it's beta. So that's a function. We call this, in the deep learning community, they call this an activation function. It's always got to be a nonlinear function, and it's, again, a way of separating, of connecting an input to an output. A convenient thing about the sigma function is I can take the derivative of sigma, and I can relate it back to the original function. If you took thermodynamics from Doug, you'll recognize some of this. If you do sigma of x times 1 minus sigma of x, and you plot, let's see, this was sigma of x, and you plot sigma prime of x, the derivative looks like that. It basically says around 0, that's where the separation is. And you separate from one side to another. So it focuses right on that region. What else can we do? We can then take this function and we can bias it and we can weight it. So if I wanted to shift it over to here, maybe to 5, because the difference between alpha and beta is maybe having 5 alanines or something. Okay, it's a little abstracted here, but this is at some layer in between where it's doing some kind of calculation. If I want to shift it here, I could do sigma of x minus 5, and I could also weight this. Say if I multiply it by 3, then I can make this function steeper. So I can make a steeper boundary between one case or another. So this, at 3, that's my weight. And the 5, that's the bias. So we're just shifting around where this separator function is. And that weight and the bias, those are going to be fittable parameters. That's what I'm going to have. So I've got this functional form sigma, and then I'm going to have fittable parameters to shift those around. All right, now I have lots of data, lots of inputs, lots of outputs. So I've got to do this many times. That's my one-dimensional function. I could have a vector function sigma of x, where each component, so let's see, sigma of x, the scalar, is that takes a real number and maps it to a real number. sigma of x, I want to take an m-dimensional vector and map it to an m-dimensional vector. And essentially we do that by having each sigma I of the vector x is going to equal sigma of the particular element. So it's just an element by element sigmoid function. Each element we're going to take and put in that sigma. Okay, so now I want to combine multiple inputs. Let's put into a network. Let's say I have 3 inputs and I'm going to draw them as circles. These are those nodes in that neural network. And so each one of these has a different input. Let's call it x1, x2, x3. And I want to feed that into a new node. And what I want to do here is I want to sum these, and I want to weight them, and I want to bias them. So what I'm going to do here is I'm going to take sigma of some weight times the first input times another weight times the second input, another weight times the third input plus a bias. And I'll take that input to my function. Now I have a fitted parameter for each weight, 3 weights for those 3 inputs, and a bias. So I have 4 fitted parameters here. And that's going to be my output. And that output will come out of that node. Are you with me? Questions? Too fast, too slow? Okay. All right, so this is my neuron. We're going to make a neural net, and we're going to have many of these neurons. Let's see, I can rewrite this as a vector W dotted with my input vector X plus a bias, sigma. All right, I'm going to call that, the output is going to be the activation of that neuron. So I'm going to call that A. This is A, which is activation. Sigma is my activation function, A is the output.
Jeff,
Yes.
could I ask you to back up a minute? The X1, the X2, and the X3, are those continuous variables? Or are they binary? Are they on-offs?
It could be either. In general, they're continuous. For the protein sequence input, that's going to be these 1 hot encodings. So there'll be 1 and 0 for the protein sequence.
Mm-hmm.
But if it were, if the feature was the hydrophobicity of the residue, that would be a continuous number.
Okay.
Mm-hmm. Okay, so I had those 3 inputs, and we put those into this guy. We can put another neuron over here and put a second set of weights and a second bias into our second neuron. So now I'm going to call this A1, and I'm going to call this A2. And I could write that as a vector equation, A1, A2 equals, well, each of those would be an equation like this. And what I'm going to do is take this equation and I'm just going to take it up, turn it into a vector equation. I'm going to have the vector sigma function, and I'm going to do W dotted with X plus B, the same thing I had here, but now X was a vector. Now I'm going to have 2 outputs, so B becomes a vector. And that vector W, now I have 2 rows of that, so W becomes a matrix, a second order tensor. And while we're doing vector notation, I'm just going to call that A and make that a vector A with an underline. Okay, so we have inputs and a layer of neurons. So this is a layer. In this case, I have 2 neurons, but I could have had 10 neurons, right? So for for that sequence where Stephen said I've got 20 times a number of residues, I have that many inputs, and I'm going to pull all those through here. So I'm going to have a lot of inputs and a lot of neurons in that first layer. I'm going to put a layer label on this. I'm going to put a superscript for layer 1. So activation of layer 1 equals that equation. And now I'm going to label these weights, layer 1, and the bias of layer 1. And you can probably see where this is going. I can put another layer in here. I should have left room. And I could take activation 1 and put it into 3 nodes in the next layer, and activation 2 and put it in 3 nodes in the next layer. And now I have activation 1 in layer 2, and activation 2 in layer 2, and activation 3 in layer 2. And let me rewrite this formula. I'm going to have activation vector in layer L is going to be sigma, a weight matrix of layer L, dotted with X plus a bias for layer L. Are you with me? Tell me this, for in this first layer, how many fittable parameters do I have, if you're with me? Or let me say this, how many numbers are in B1 and how many numbers are in W1? If you've got it, give me a show of hands. How many numbers in B1? See some hands up. Give me a number of digits. You might need 2 hands. All right. I see it correct from several people. I see a lot of people who are a little lost still. If you need to stop me, stop me. All right, yes, there's 2 numbers in B1. There's the bias for activation node 1, and there's bias for activation number 2. So this is a 2x1 vector. And how about W? What are the dimensions of W? I see 6 parameters, yes. This is 2 outputs by 3 inputs equals 6. So the total number of parameters here is I have 8 fitted parameters coming out of the second layer. All right, now I make many layers, and I'll leave it to you as an exercise to find a formula for if you have many layers, how many parameters do we have? All right, so. Ooh, I missed one thing here. The X here, I'm going to change this X in this general formula. I'm going to make that A from layer L minus 1. I'm going to take the previous layer as the inputs, and I'm going to let A0 equal X. So I'll initiate my column with the inputs. All right, so where are we then? If I had A1 dotted with X plus B, not A1, sorry, W, in layer 1 dotted with X plus B1, and I took the sigma of that. That's going to give me A for layer 2. Then if I weight that and add a bias, for the second layer, I take the sigma of that, that's going to give me what comes out of the second layer. And if I then take that by the third layer, add the bias of the third layer, take sigma of that, you get the idea. These are nested within each other, and you have functions within functions within functions. And that whole thing I'm going to call f of x our giant function, and it could have many layers. It generally, if it's over 4 or 6 layers, they start calling it a deep network. In proteins, this is common to be 50 layers deep. The deeper it is, the more structured your information, the more parameters, the more stuff it can learn, the longer it'll take to train. All right, so next you need to say, how are we going to find all of these W elements and all of the B elements? And the way we're going to do that is by taking this function, and we're going to look at how that compares with the actual outputs. So I'm going to subtract these and take a norm. So Y is this is the experimental, what do you call this? You call this the ground truth, is the deep learning term for it. That's what's actually correct. This is the structure as determined by crystallography. F is our model, and X is our inputs. And then we're going to take a norm. There's different choices of how to take a norm, but in general, you're taking some kind of distance and squaring it. We're going to do this for each. We're going to have many, many data points, many sequences, and many structures. So I'm going to label those with an I. X of I. That's coming out okay. So and I'm going to sum this error up over each data point n and divide by n. So n is the number of, these are my data points. And this, I think one of the things I'm going to be happiest for when we get back on campus is to have boards and boards and boards I can fill at the same time. This is called our cost function, or the loss. It's how much error there is between our model and the experiments. And this is essentially, C is a function of the set of all of the W's for each layer and the B's for each layer. Ah, that board's getting messy. All right, where are we before I run out of time? Okay, so then the game in machine learning is to take this thing, you want to adjust all those W's and B's to minimize C, the cost function, which then gives you the model, the f of x that you want to have. I'm not going to go deep into this. There's a whole bunch of ways to do this. It's similar to anytime you're fitting, your least squares fitting, and you're finding parameters to minimize some kind of penalty. The most common one is to take, because you have tons of data, is to take one data point at a time. One of the nice things with that sigma function is it's differentiable. So you can take this giant function f of x and you can actually calculate df d some parameter. As I change a parameter, how is the loss function going to change? And you can calculate that analytically. And in your code, you can do that. So you can find those gradients. You can use Newton's method and find a minimum. Okay. All right. So many parameters. How do we, so it seems like a little bit of a cheat, which it is. We're learning from these data. One trick I wanted to mention is if we take our amino acid sequence, I don't know. If we take our amino acid sequence, one thing we can do, instead of operating on all of that at once, you can operate on a few amino acids at once. and feed that into your convolution, your W, and your bias. But you could do the same thing with different windows of amino acids. You could take residues 2 through 5 and 3 through 6 and 100 through 103, and you could apply the same matrix to these. So what you're doing is you're reusing your parameters in different places along the sequence. This is what's called a convolution. And in a convolutional neural network, instead of having to have weightings on all of the inputs, you do it on a small window, and you move that window along, and you do the same weights on that whole window. There's other tricks you could do. Instead of doing consecutive ones, you could do every second or every third residue and accumulate the data that way. All right. Questions about the math? That's the heavy math. I want to go to some protein stuff and some implications. So we started this in the lab with a summer intern. I said, I don't know anything about deep learning. I don't want to do that because I like physics. But this summer intern who came from Georgetown and is now at Google took a summer and actually got some results very quickly. So I want to show you the network he put together. And then Jeff Ruffalo, who many of you know in biophysics, took that over and made it into something that wasn't just a summer intern project, but that worked really well. So this is a program we call DeepH3. And the problem we chose to work on, you all know antibodies have this structure with 2 light chains and 2 heavy chains. And if you look at the business end there where it's going to bind an antigen, this is an antibody, you have 2 domains. You have the variable domain on the heavy chain and the variable domain on the light chain. And those bind to an antigen. And the structure of an antibody is mostly conserved. It's mostly the same. But what's always different is the so-called complementarity determining regions. There's 3 loops on the light chain and 3 loops on the heavy chain. And your immune system makes these antibodies diverse by mixing and matching different gene segments. Those gene segments are called the V and the D and the J. And it just so happens that the H3 loop is at the intersection of those 3 segments. And so the combination of those segments gives you a lot of diversity. It's not predictable. It's very random. And every antibody has a different H3. The other loops are conserved. There's different classes and we can plug those in based on some sequence homology and have a good approximation of their shape. But the H3 is always different. So I like this problem because it's different than the canonical problem of take a sequence and find the fold, which everybody in CASP is doing, and there's hundreds of groups there, so I don't need to compete with them. This is a small local feature, and what I really care is getting the details of the loop conformation right. That's what really determines how it binds an antigen, if I can engineer it, make it bind it tighter, make it change the specificity. So I really want to hone in on those details. It's been a hard problem for us to do. We've worked on it for many years, and let's see if deep learning can do it. Also, there's no evolutionary data there. That's not completely true. If you take a repertoire from an individual patient, you can see that loop evolve. But in general, if you just have a structure in the PDB, you're not going to have other homologs that you can find in a database. You have to have the individual who evolved that particular sequence. So that's different than in CASP. The CASP, people use a lot of evolutionary data and couplings between sites to help drive their predictions. All right, so we had a training set of about 1,400 structures. That came from the SABDAB database, which is done by Charlotte Dean's lab. What you do with that set is we divide it. We use 95% to train our network and to teach it those parameters. And then we use 5% to test. That's a little lower than is usually done, but this is a small set. There just aren't that many structures of antibodies. We weren't sure if this was going to work. We weren't sure if we should train over all structures and then predict antibodies, or if we should just train on antibodies. But this is what we ended up doing and ended up working. The other thing we had is a separate 50 antibodies that we'd been using to test Rosetta antibody, our Rosetta-based predictors. And so we just took all those out of the set. So they're completely separate and don't have anything to do with it. And then what? We took this giant structure, we fed them through the network and out of the network and trained the network and came up with some results. Let me show you the network structure. And let me show you another detail of the outputs, because these are some key things to get it to work. Okay, so we take, this is the CNN, which is a convolutional neural net for protein folding. And I won't take credit for this architecture. Jinbo Zhu is the first one who published on this architecture. He did it for protein folding, I think back in CASP 12, and did okay. And this is the architecture that AlphaFold took and used in their most successful stuff. So the first thing you do is take the sequence and you hot encode it. You take your 20 bits and your 1 to n res, and that's your input. We feed that through a neural net, like we just described all the math with that activation function sigma. Actually, they sometimes use some different activation functions. We use a different one called ReLU, but those details aren't real important. It does the same thing. And you put these through several layers. Now the output I want to have, actually, if you put these through 7 layers. So let's put a ... in here. The output I want to have, if that's going to show up, is that distance matrix where I can see helices and see strands. And I especially want to see contacts between the H3 loop and the L3 loop and other things. So that's what I want to get. Now this data is 2-dimensional. This data is 1-dimensional. So there's a little bit of a trick here where we take this 1-dimensional data and we duplicate it, we copy it. So I've got about n-res long here and about n-res long here. And depending on how many nodes I can put in, these data sizes can shrink or grow depending on how many neurons there are and connections. But I take this and I put it together to make a 2-dimensional matrix. Okay, so it's just pasting 2 matrices together and basically sticking them together. Then we do layers, and now my input, if you're losing track of the tensor ranks and all that, that's okay, I am too, but now we're gonna take that matrix as our input, go through a layer and get a matrix out. So that's going to go into a matrix and then into another big matrix, etc. Here's where some magic happens. They're going to, Jinbo Zhu and Jeff Ruffalo, we use about 50 layers, 51 layers in DeepH3, and then end up with this square. And from there, the last layer, I want to actually predict those distances. In DeepH3, I have 6.4 times 10 to the 6th parameters. So what curve can we fit with 6 million parameters? This one, okay. So yeah, okay, so this output is the C beta distance. C beta distances are nice. Yeah, okay, here we go. But there's a few tricks. One of the tricks is to use labels or outputs that are directly coupled to intermolecular forces. Okay, so you might ask, why don't we just output a vector of phi angles and a vector of psi angles? That would tell you the fold if I knew all the angles, right? Well, here's why. Let's say I have this protein and I'm looking at a residue in the middle and I'm trying to predict phi or psi. If I change that phi or psi, I have this lever arm effect that's going to move everything in the protein, right? I may have these two residues at the C and N terminus interacting, and they're very dependent on every single phi and psi. So it's a highly coupled problem. It's going to be really hard to find the right values of phi and psi. Now, what I know from my physical intuition is that this interaction, if that's two hydrophobic residues, they're going to fit together and their shapes are going to match. That's where the information is. That's where there's high quality information and it's decoupled. Well, those two residues would show up, there in this grid where it's, at the distance between those two residues, but it's not just the distance that's going to matter, but the orientation. It's got to have the right orientation so that the side chains can fit. So we define, and we took this from paper from Yang and out of David Baker's lab, and Sergei Ovchinnikov, who's now at Harvard, did a lot of this. If I have an alpha carbon, a beta carbon, beta, alpha. So the simple thing that everybody's been doing since CASP 13 is looking at that distance. Well, you can also say, how are these rotated relative to each other? I'm going to call that omega. You could also say, where are these backbones? And the backbones, you can move around by swiveling this torsion angle around and by changing these bond angles. So we're going to call this bond angle phi, call that phi ij. This bond angle I'm going to call phi ji, because it's the opposite way around. This one I'm going to call theta ij, and this one I'm going to call theta ji. Are you following me? So I'm starting from the C betas and I'm looking backwards and setting all the angles that puts the backbone in the right place. And once that's set, that's going to tell me which way the two chains are going. In fact, if you've been counting 1, 2, 3, 4, 5, 6, I have 6 parameters, 6 geometric parameters there. 6 is exactly what I would have to know to say what are 3 translational variables and 3 rotational variables to tell me how are 2 coordinate systems related. So if I have a coordinate system for each amino acid, it's going to exactly place those 2 somewhere else. So in DeepH3, as in TR Rosetta, which is from the Yang paper, we're going to have several outputs. We're going to have the phi matrix, the omega, the theta, the distance, 1, 2, 3, 4, 5, 6. That's it. And the phi's and the thetas are asymmetric. So what's on the top and bottom of those matrices are different. Still with me? All right. So that's the general story. That kind of gets you up to date through CASP 13 and through our first DeepH3 papers. Am I seeing a hand for questions? Or I can pause. I have a few advanced tips in what's changed since this last CASP and a little bit of philosophy about the outlook. But okay, Doug, Martin. Let's go to Martin first. We'll start with students.
Yeah. So I was wondering, so is the training data and the output for DeepH3, is that like the free loop segment, or is it the antibody crystal structure itself? Because I'm wondering whether, if it's the antibody crystal structure, whether DeepH3 like learns the constraints of the loop, in that like it knows that it always has to be bent no matter what. Like thermodynamics might say that it should be like stretched out or something, but if it's always seen antibody crystal structures, the 2 loop ends will be fixed.
Yeah, we train it over our range as we go from, actually go from residue 1 to n in the heavy chain and then 1 to n in the light chain. So we actually use all residues of both domains, both variable domains. Why? Because I want distances, when I have the heavy, and here's that H3, I want distances over to those light chain loop residues and distances to the other loops in the heavy chain. And then, I don't know, it's also learning about residues and their distances and their energetics by looking at both domains. Although if that was the case, I could have used all protein structures too. Yeah, that's what we do. I'm not sure I have a great justification for, for that being the best. Doug.
You can ask Ben. Ben's a student.
Oh, Ben, where's Ben?
Hi. So one of the things that I've heard about when people discuss deep learning algorithms is that they're somewhat like a black box. And obviously when you have like 51 layers of multidimensional data being shoved through the algorithm, and you're getting out these numbers that correspond to physical things. Like you said earlier, like as someone who's more of a physicalist, like you want to be able to understand what the algorithm is doing, right? So is there a way to work backwards? So once you have like an AlphaFold 2 that can accurately predict structures based off of sequence, to understand what that's doing based off of the parameters?
Yeah, that's a great question. They call it physically interpretable AI or AIX. I don't know why it's an X, but explainable AI, that's the X. Yeah, so I can take a look in any one of these nodes and I can visualize it and I can say, what are the parameters and what are they doing? And one thing I could say is, in this node, I could search for, are there any nodes where every time I put in an alpha helical protein, it's a 1, and every time I put in a beta strand protein, it's a 0. And so you can actually find a node where you go, oh, right here, it's identified the secondary structure, right? So you can look for things like that if you know what you're looking for. And people have looked and found some of those patterns that we know about, but it's obviously finding other patterns that we don't know, and I'm not really sure how we'll find that. I will mention too, this is one of the bonus things that came on the last couple of years. There's this thing called unsupervised learning, where you don't give it the outputs, you just give it the inputs. So you give it a bunch of protein sequences. And what you do then is, to train it, you delete some of the sequence and you ask the model to predict what goes back in there. So it'd be like if I had a missing in a sentence, your brain fills in what that missing element was, right? Because you pattern match that, or you're reading and you see a strange word, you have some context clues and you figure it out. That's what you're asking the machine to do. Just take a bunch of sequences, and then as you delete parts, can you fill it in? And when they do this, so that's called one, it's called unsupervised learning, because there's no labels, you're not telling it what the right answer is, but it learns the structure of the data and how they relate. So they do this with language all the time for translation. And so Jeff has done this, and Pooja Mahajan in the lab, they've done this, and we're looking at, could you identify when the H3 has a kink to it, or when it has more of a straight beta strand feature. So we can look for some features and see what it learns. You can see that it learns, even just giving it sequences, it learns to separate out mouse antibodies from human antibodies. It learns to separate out long L2 loops from short L2 loops. So it finds that structure in the data without telling it, here are the structures. So yeah, this is one of the puzzles. This is one of the big philosophical points, is what do we do with this, right? It's very, after you train it, it's very fast, whereas Rosetta antibody takes us 300 CPU hours. DeepH3 is done in seconds, right? After it's trained, once it's trained. And it's fairly accurate. I didn't show the accuracy. You can look in Jeff Ruffalo's paper, but we're beating Rosetta antibody a lot of the time. It's going to be useful for structure, for design, for docking. Now we kind of have some proof of concepts, and we have to adapt this structure to it to tackle all of these other problems and use the structure that we know. But then yes, the understanding, I don't know, with the, with putting these geometries in, our physical intuition tells us how to better train, how to better coach the network to learn. And then hopefully probing inside the network, maybe we learn other patterns that we haven't seen yet. All right.
That was great. Jeff, I have a question also.
Yes, Mario.
What do you gain by the extra 5 parameters? The distance between the beta carbons, if you use distance geometry, any of the old programs, you get a structure. So, so if you were saying, just use that, what do you get? Less accuracy, too many structures to the side. What is the advantage? Those parameters, especially omega, have the same problems as phi and psi, which is if you miss them, they'll just don't make the ends be right. So there are constraints that are being used, but how bad is it if you use only D? How much do you get?
Yeah, so what you can do is look at that loss function, and as you train over epochs, they call them, and you train and you train, and you see the error goes down as the machine learns. And if you added these extra geometries, you can see that there's less loss and it learns better. So you do get better answers with this empirically. You're right, if you had all the C beta distances, you could regenerate the structure if you had them right. The thing is, by giving it these clues, here's my theory, is that it could potentially learn an energy function from this. If you know the distances and the angles, that should determine the energy. So it could in those middle layers. Now it doesn't have to, because I don't know what it's learning. There's many ways to fit a function correctly, but it could be learning the structure of the energy function. What happens when 2 residues with backbones here come together? Do they make a hydrogen bond? Like with a certain distance, they may or may not make a hydrogen bond depending on the angle. So this is me telling it what to pay attention to. So I think about this like if I'm coaching my kid how to hit a baseball, I might tell him how to hold his hands, and I tell my daughter where to put her feet and tell them to watch the ball. And this is telling the network what to pay attention to do the task right. That if you look at these angles and you structure it the way physics should structure it, maybe it could still learn it with just the distances, but it might take longer, it might be more difficult, you might not converge as fast.
Now, one thing also to realize, and that goes to the question, the previous question, which is to get from these parameters to an energy function. For the energy function to be believable and understandable, you still will have to use R6, R12, and you still will have to use a partial charge or something like that. So it will be interesting to see how it ends. Yeah.
Maybe I just need a function of distance though. You know, I can do G of R and I can recover all my thermodynamic properties. So maybe I just need the function as, it may not match a 612 potential. I mean, it should, in principle, have some, if all the physics we've been doing is right, it should match some van der Waals and electrostatics and hydrogen bonding, but it doesn't have to. It doesn't have to.
No.
Yeah, and I guess I would even add to that. I mean, the 612 potential is not that miraculous either. I mean, there's other functions that would get it right. So
That's right. That's right. It's not even theoretical, right?
So Jeff.
Yes, Doug.
Ben asked the first half of my question. I thought it was a great question. But related, you said you have almost 10 to the 7 parameters, adjustable parameters. And if I sum all of the elements in each of your matrices, it's a lot less than that you're predicting, but the inputs are a lot less too. So the coefficients that you're fitting are horribly underdetermined, right?
Mm-hmm.
So does that, how big a challenge is that in terms of doing this thoughtful AI you were talking about? If there must be a lot of different solutions with very different coefficients that give you the same loss function?
Yes, I'm posing it as a minimization problem over the parameters.
Yeah.
So that is well posed, but there's multiple solutions. People usually do it stochastically, pick different training points at a time as you're optimizing. And the trend now is to train many models. and to run all your models and take averages or some kind of consensus from many models. So yes, it's underdetermined. You'll get multiple solutions. So yeah, even if I determine the energy function in one of them, it might not in the other one.
Mm-hmm.
Yeah,
Right.
that's a good observation.
Jeff,
yeah.
can I also perhaps follow up on that?
Yeah, but let me first acknowledge you. Jeremias is a new BME professor, not so new anymore, but Jeremias has been our muse in the lab for all things deep learning, because he's our deep learning expert. So yes, Jeremias, please.
No, I think I've been the one learning the most out of all this project, because it's taken me back to biophysics and other things that I hadn't looked in a while.
Very fun.
Anyways, going back to the point that was just raised, that's exactly a terrific point. You're saying, hey, I have these many parameters, I have way fewer samples. I can fit anything. I can fit noise. How on earth would that be any good for prediction? And that's a totally accurate observation. Now, turns out that these models oftentimes, most times, find good solutions. And by good solutions, I mean solutions that generalize. So even though you could be fitting to random noise, turns out that the solutions that you're fitting are actually those that perform also very well on data that you have not seen. Now, how's that possible? Well, that is one of the mysteries that people working on trying to understand these things a little bit more formally and theoretically are still working on. One partial answer is that actually many of the tricks and add-ons and hacks that we typically employ when training these things, in fact, adds quite a bit of regularization. So for example, in some settings, it is understood that using stochastic gradient descent to minimize this very non-convex function has very strong regularization properties. And in some cases, we know precisely how to characterize these solutions, and we understand that, okay, these solutions, even though there are infinite ones, the solutions that are found by gradient descent are actually the ones that you would like to find to begin with. They are solutions that generalize well. Other things, for instance, dropout, which are some other techniques that are used, oftentimes also introduce sort of implicit regularization that are increasingly demonstrated to have nice properties. So as you see, all of these tools came out from massive engineering efforts, but they were, it's been a huge experiment. All these tools were developed basically in a sort of evolution of ideas of sorts, and the ones that were proven to find very good solutions have evolved, and only now we are starting to understand them from a more principled point of view.
Thank you. Yeah, that helps. Yeah, the dropout trick you actually remove points and force the network to still do as well is what you do as you're training. You drop off points as you go, and that forces it not to just overfit to individual points. So there's, yeah, the other secret to this, Jeremiah is right, we're building on a lot of work by the computer scientists. These tools are surprisingly easy to use. You just import TensorFlow or import Torch, and you've got them, and then you can build these networks pretty quickly. So it's a tool I think a lot of biophysicists may want to at least have some basic know-how to be able to plug in when you want to fit some data. I see Steven's hand up.
Yeah, no, this is great. Thank you so much for kind of really explaining it to a layman, which was fantastic. So I just want to make sure I understand properly. So once you have the model, it is deterministic, right? Like it is just a function, but I guess training is going to involve some degree of like guesswork to figure out which parameters to model and when, right? So that's where it becomes more.
Yes. Training it takes a lot of iterations back and forth. So Google DeepMind, gosh, what do they have? Jeff would know. It's something like, they had something like 128 of these, I forget what they're called, K2 GPU processors or something. It was equivalent. People were trying to calculate how many millions of compute time that was worth, but they wanted lots and lots of computers to crunch all these data points and try to find their things. So training can take forever. That's one of the big advantages that Google had over us, over the whole academic community.
But I guess like the other thing that maybe I'm curious about and also point out that the training is also like stochastic, right?
And training is stochastic because you can't load in the whole PDB and fit to all of it at once. So what you do is Pick one structure and see what the loss is and adjust your parameters. Pick a different structure, see what the loss is, adjust your parameters. Yeah. So that's
you think involve like some Monte Carlo type of way so you can get out of minima and sort of pop around. I can imagine with like
I think when you pick different training points, you fit to one enzyme and the model works okay, but then when you pick a different enzyme, that pulls you in a different direction. So that's essentially changing the landscape as you go when you're fitting to different places, as I understand it.
Cool.
So this is a completely unsupervised model. Would you ever use a more supervised model? What would be the advantage or disadvantage between the two?
Yeah, so a supervised model, when you know the answers and you want to get this answer out, that's what you use. The unsupervised model, I'm still figuring that out actually. What we're going to do, so we're training this unsupervised model, and then we're just going to stick it in as another input. We're going to take the sequence through the unsupervised model. It gives you some representation, and then we stick that in as another input that'll have some structure to it. In language, you get this model of English and a model of German, and now you can translate between because they're connected to each other. Or you can, you've seen this in your email, you write half of a sentence and Google knows what the rest of the sentence is. That's that same model. It's learning it and it's making predictions. So design, the unsupervised model is going to be great for design. So if you have a lot of a protein and then we cut out a segment, we could ask the model, what kind of loops can we stick in here? What kind of loops does the model think would fit?
So that's more like NLP kind of stuff with like BERT and stuff like that.
BERT, yeah, BERT is the model that we're using.
Okay.
Yeah, and they've been trained now. The biggest BERT, I was just looking at this, was has been trained on like 1 billion sequences because they're these giant, the big fat sequence database or something it's called, has 2 million sequences. So they've trained with this giant pile of things.
Oh, thank you.
So I have a quick question. Let's say I get hold of the sample that they've returned from the asteroid in Japan, right? And it's got plenty of amino acids. And can you tell me anything about what sort of structures you might fold up into? You know, if I just give you the input from the experiment, from the sample return.
Say again, if I give you, if you're...
Do you know about the sample return from the
Mars?
asteroid?
Okay, yes. So
Mars,
Mars,
no, not Mars. Not Mars. It's from a comet, an asteroid.
Oh, right, sure.
Yeah, Hayabusa or
acids?
something. Yeah, and so let's say you get that data. It's all simple, you know, it's not complex chemistry. It's just a bunch of amino acids and a few other icy things. What can you tell me? What can I put the data into your enormous computing machinery, and can I get out anything sensible in terms of predicting what this might fold up into?
Based on those CASP results, if you put it into AlphaFold 2, not my model, but the AlphaFold 2 model, you'd have a pretty good chance of having a model that's accurate to 5 angstroms about what the structure of that protein looks like. That's the current technology, and that's if you asked me 2 years ago, I would have said, yeah, you know, maybe a third of the time or half the time you might get a decent model. But based on that DeepFold model, you're more than likely to get a good structure from a sequence alone of a completely new, never before determined protein sequence.
Well, it seems to me that's one of the simplest experiments you can do and one of the most important.
Yes.
Well, I don't know. You guys want to cure, you know, humans, etc. But just in terms of fundamental understanding about life, we're going to get the data shortly. You could probably do some sort of models like that. Yeah.
As far as the fundamental, I mean, this is a Nobel Prize level question. Sequence to structure, we've worked on it for 60, 75 years.
Yeah. And
Mohammed El-Karishi said he didn't think this was going to happen for another decade, but he said at this point, he said the scientists should be done. If you're a young scientist, why would you go into this field where the Nobel Prize is already given out? He said at this point, it's an engineering problem. And I said, thank you very much. I'm an engineer. Now I can figure out, can we cure diseases? Can I tell, you know, my colleagues at the med school that have two interesting proteins, how do they fit together? Or if I mutate it, what's going to happen? So all of that work of using it and applying it is wide open now. And you know, the models are very specific. All it does is fold a structure. So we've got to find a way of generalizing it. You know, MD at least has forces and such, so you can apply it to different things and ask different questions. But the only question you can ask with this model is, what does it fold into? So we need to figure out ways of generalizing this and using it in different ways so we can solve all these meaningful questions that the biophysical community and the medical community and the biological community want to know about.
So I have a question.
Yeah.
When Alpha 2 was being trained on the PDB, did it identify any incorrect structures?
I don't know. They didn't say, and probably.
It'd be nice if they listed them now.
Yes, it would. It would. I'm being told that somebody else has the room and they need to clear out the room before. I could hop out and jump on my laptop if people want to stay, or you can email me and contact me with other questions. This is super fun.